Welcome to Bachem and our webinar on trends in peptides and oligonucleotides, choosing a partner CDMO. My name is Daniel Grotzky, and I will be introducing you to today's webinar. Before we get started, a word of disclosure. We are using all the opportunities the digital world is offering us and pre-recording the first segment of this webinar for all of you. You will have the opportunity for a live Q&A after the pre-recorded segment. Your main speaker today is Tycho Leifels. He is Group Leader Product Management at Bachem AG here in Bubendorf, Switzerland. I am Head of Group Communications, and you can see from the picture I have shaved off my pandemic beard already in a hopeful sign that we might meet face-to-face sometime in the near future. Before we get into the nuts and bolts of the presentation, a few words about Bachem so you know whom you are engaging with today. Bachem is a CDMO, so we are a supplier for the global biopharma industry. We are a very specialized company specializing in the manufacture and services around peptides and oligonucleotides, two sets of very complex molecules that are used as APIs in a number of medicines. We focus on long-term partnerships with our customers, and our main purpose really in the world, along with so many others, and as we've seen particularly during the pandemic in a very important way, is to help patients, help through our customers to get medicines to everyone in need across the world. We are a very long-term oriented company, so our planning goes into the next 10 years, and we want to build our leadership in peptides further out, as well as establish a increasing presence in oligonucleotides. As a leader in our field and with these strong growth ambitions, there are really five main elements, five strategic foundations that play a critical role in how we conduct business at Bachem. The first one is about people and culture. We rely on experts in the space of tides, and so this is, by definition, a very strong people-focused business. The second is innovation and technology. We really aim to work with our customers to get the best possible solution, the most modern solution in terms of manufacturing the products that we bring to them. The third one, and increasingly important really, is sustainability. We owe it to our customers, we owe it to the patients whom we serve together with our customers to make sure that the impact that we have on the environment is as small as possible. Then customer centricity plays a huge role. We are a supplier to the industry and really reliant on very strong long-term relationships with the customers and clients whom we serve. And finally, Bachem has a long-standing tradition of quality and efficiency in everything that we do. And again, something that we really owe to those people who use the API we create in medicines that they might be taking each and every day. We are a global company growing fast over the past few years with presence across both Switzerland and the United States and additional sites in the UK and Japan. So you might bump into us in any of these places and even further across the world. We focus on products and services in three categories. The first one is research and specialties. That is the area where we have customers, particularly in the research space, but also in cosmetics or diagnostics. We usually create small quantities of molecules there. And then the second one is CMC development. There we partner with our customers in clinical trials where medicines are being developed. And then finally, the area of commercial API, where we supply large quantities of API for both generics and originator products, NCEs. So with that, I'm very happy to hand over to Tycho, who will take us through three major points. He'll share some trends in the peptide and oligonucleotide market with us. We'll be speaking about how we think about partnering with a CDMO, what are some of those things that are significant there, and then also talk about large-scale production and capabilities there at Bachem. So without further ado, I'm very happy to hand over to Tycho. Tycho, please come over and take the stage.
Thank you, Daniel, and welcome also from my side to this webinar. It's a great pleasure for me to share some insights on the trends in the peptides and oligonucleotides market with you. I will tell you why I think that partnering with a specialized CDMO will be part of the success for your complex APIs. And I will also cover some aspects of what it means to produce such products in large scale. Peptides and oligonucleotides share the same principle. The coupling of small basic building blocks leads to a giant number of different products that can be produced. This was, after all, the winning principle after thousands of years of evolution. They are the substances that the way we are today. Using molecules that are closely related to natural substances can have big advantages in pharmacology. The body has very efficient ways to metabolize those products. The result is that you can predict very good how these products react in the body, much better than for conventional chemical drugs. And because they're highly selective, the drugs are always very effective and can be used at very low dosages. PubMed is a database that captures scientific publications. We have searched this database with the keywords peptides and oligonucleotides. And the growing number of articles is an indication that the scientific interest is really increasing over time. The graphs, however, also show that there is a difference between peptides and oligonucleotides. Peptides already were a thing in the 1950s, and with oligonucleotides they started much later. And you can even see a difference when you look for subterms like antisense, which started in the late 1980s, or then aptamers, middle 1990s, or then siRNA that started in the year 2000. And you also see that the scientific interest is directly reflected in the pipelines of pharmaceutical companies. 78 peptides are already commercially available, 314 are in clinical development, and 848 are in preclinical stage, many of them even in late clinical stage. For the oligonucleotides, it's still a bit less. It's 14 that are already launched, but there are 800 in the pipeline at different stages. 3/4 of them are still in preclinical stage, but from the ones that are in clinical stage, again, a large number is already in phase 2. The peptides are already used in a broad range of therapeutic areas. For oligonucleotides, in clinical research, this is or starts to be already true as well. You will find the usual suspects of the big indication areas like metabolic disorder, oncology, CNS, or cardiovascular disease. The first oligonucleotide therapies were targeted at rare disease, orphan disease, often not treated by other treatment options. This has started to change, and the shift towards therapies for common disease can be seen. So in those areas, the new therapies will go head-to-head with existing therapies. They need to be compared in their effectiveness and their safety, convenience, but also on cost. The therapies are also evolving. Higher efficacy, better tolerability, and convenience are key goals. For the drugs, this is often translated in a more targeted delivery, prolonged activity in the body, self-administration, if possible even a non-parenteral device without a syringe. And this very often has also implications for the API, the active pharmaceutical ingredient. We see more side chains, conjugates, and non-natural amino acids in peptides, like the example liraglutide on the left side. In oligonucleotides, like inclisiran on the right side, you find modifications, and in this case, it's modifications on the sugar, modifications on the phosphate, and even the base pairs. So it's getting more and more complex. And after the recap of the market trends, let me now try to answer your question, how a CDMO can support you to get your drug to the market more efficiently. Of course, you can always also develop and manufacture an API yourself. The advantage clearly is that you have full control over your intellectual property and full control over the whole process. However, I'm convinced that those are by far outweighed by the advantages that you have when partnering with a specialized CDMO. The value that we bring to drug developers comes from satisfying their needs. The first challenge for us is that the needs really differ, from startups to big pharma, from innovators to generic companies. Yes, they all want the lowest possible price, the highest quality and reliability, and everything delivered yesterday. In the details, the needs, however, vary quite substantially. For a university research group or a small startup with just a few people, this is very often their first research project, and they rely heavily on the expertise that we bring with 50 years plus on expertise in CDMO activities. And they also rely on the dedicated project managers that will support them. This experience is also important for the large pharmaceutical companies, and we have the largest ones as our customers for long term. And this is the case because they also want the reliability, the experience from a partner like Bachem. Their focus is, however, more strongly on our flexibility to adapt to their processes. The needs that a potential customer has also changes a lot during the life cycle of a product. Speed and flexibility are key in early development. The ability to manufacture the desired batch size and a perfect regulatory dossier become very important during later development stages. Then, towards the launch, secure supply chain, compliance with all regulations, and the lowest price have the highest priority. Depending on the focus areas of the customer, sustainability, as well as planned life cycle management, or possibly even own IP, can be a main point of interest. And once the product becomes generic, prices tend to fall, volumes go down, and that's an issue we need to address. And sometimes the product is then divested to a different owner, and we have to support this as well. Let's now take a step back and go back and imagine you start with the development of a new product. At Bachem, you find a catalog with a lot of peptides readily available. And we also can produce your sequence of choice as a custom synthesis. Our experience allows us to deliver even complex products with superior quality. Of course, you can also do this yourself, but we have a large number of synthesizers. We have the experience, so we probably are faster, at better quality, and less expensive. Major success factor afterwards is how fast you're able to reach the market with your product. Once you have filed for a patent, the clock starts ticking, and very often the competition isn't sleeping either. The way it's done is usually by squeezing the clinical phase as much as possible. Very often, clinical phase 1 and 2 are squeezed the most, sometimes even done in parallel. This is challenging for an API producer because, as you can see during this period, there's a lot of work packages that need to be completed. And you want definitely an experienced partner who can do this as fast as possible. Afterwards, the GMP development needs to progress at high speed as well. And you need to scale up. But very often, you don't know in an early phase to which size you want to scale up. This is determined by the clinical studies that you're doing. And those clinical studies, you're not sure what the outcome is. It might be a step back, or it might be the development in a different indication. So very often, it's a way we called Warte, Säckle, Warte in the Swiss Army. It translates into wait, rush, wait. We hated it, but it's unavoidable. Yes, are you willing to wait? Are you ready to rush? It's something that really shows the advantage of working with a CDMO partner who has the capacities at different sizes readily available, where you can choose from different sizes instead of deciding for one. The analytical requirements of a complex API are also very demanding and increased in requirements over time. The difference between your desired product and an impurity are very small. Very frequently, it's just a change in one amino acid or a minorly truncated oligonucleotide. So safely separating and identifying those impurities can be very demanding. A lot of experience, but also the latest technology can dramatically improve the outcome. And for complex products, you also need to characterize very often dozens of those impurities. A great benefit for us is that we have our facility in the UK that is specialized in manufacturing small quantities. They can quickly supply impurities and reference standards without involving any additional third source. And the long list of capabilities that is listed on this slide also shows you what changed over time, because the requirements from regulators, they have increased dramatically. Once you're ready to register a product, you want the perfect file. But you also want to spend as little as possible. Our registration specialists, they know exactly what to do for complex APIs. They know what is nice to have and what are absolute musts, because otherwise you will run into questions from the authorities that you can't answer without repeating work packages. The end is a delay. And once the registration has gone through, you want to launch the product ASAP. But then think again, you need to produce it as well. And once again, it's this wait, rush, wait. When producing a peptide, usually you have some lead times. Producing it requires often 2 months, because you have to couple one amino acid after the other, purify it afterwards. After those months, you do the finished dose product, and for a sterile product, the release will even take months after having manufactured it. So, being able to produce on time is very important. Of course, it would take the same time if you would do it yourself. The advantage that we have is that we're not producing one peptide, we're producing a lot. Many different peptides use the same building blocks, so we have them even on stock, and we know from what source we can order them at high quality. And that's the big difference that we bring to the table that adds flexibility and speed. And talking of the supply chain, sourcing starting materials is one way of securing the supply chain. The steps we take to secure the supply chains are also having material on stock, but it also includes having more than one supplier, knowing the quality of the products and suppliers, and being able to manufacture them ourselves if required. And equally important as a solid, high-quality process. Nothing is as bad as not being able to produce because you have failed an audit. Bachem, we did not get a single critical observation like a warning letter during the last audits in the last 10 years. In most inspections by FDA and Swissmedic, there is not even a Form 483 being issued. And high-quality process and automation will also help you to have a really consistent product. And manufacturing them to stock means we're even flexible enough to supply you another batch if one batch should fail at the finished dose stage. The next thing I want to talk about is innovation. I think this is the key driver for a competitive advantage and for sustainability as well. Let me give you a few examples. The first one's continuous chromatography. It's a technology that allows you to use smaller equipment. You can go with a 60 centimeter HPLC instead of a 1 meter column. But you also reduce the amount of organic solvents, while at the same time you're increasing purity of the product. It doesn't come for free. The development is much more demanding, but the result is very convincing. As we talked about sustainability, that's one way. You reduce the amount of solvent that is required, and that's, of course, also good for your wallet. A second example is molecular hiving. That's a technology between the solid phase synthesis and the liquid phase synthesis. You're coupling your product to a support, but this one's in solution. Again, a possibility to have a product that offers some advantages like larger scale, but also a product that can be produced without CMR. So carcinogenic, mutagenic, or reprotoxic agents can be completely left out of a synthesis using this mechanism. And if you're interested in those, I would invite you to other webinars that were done on those two topics. Automation is another key innovation. Automation is not just the answer to the challenge that we manufacture in Switzerland and California, not exactly the places that come to your mind when you're looking for cheap labor. Baloo, the robot on this picture, works for us all 3 shifts 24/7 and needs very little breaks. So the obvious advantage is higher speed, lower cost. But higher flexibility is as important. You do not need to consider shift ends or weekends when you're planning your synthesis. And the less obvious advantage is that you will also generate much more information about your process. And that's not just a robot, but it's the process in itself. You have a much tighter control. You have data on temperatures, pH, whatever you want every second, every minute, the way you need it, instead of having it in a manual protocol every hour. And you can control your process in ways that you didn't even know were a thing before by using artificial intelligence to improve this. And once you have nailed the optimal process, you can repeat it spot on every single time. Being able to produce at large scale is very important to timely deliver the quantities required. Scaling up also has a very favorable effect on price. However, scaling up also means you need to increase your batch size, you need to increase the starting materials, and some of the technologies like HPLC or lyophilization do not scale that easy. So if you want to have lower prices, what is much more effective is to improve your process. When I did my PhD, I was happy if I had a yield of 80% for one chemical reaction. I even got suspicious when I read a paper with more than 95% yield. Very often we joked in the group that they probably also included the magnetic stir in their result. Now, if you have a yield of 80% for a single step in a peptide synthesis for a 50 mer, both for coupling and deprotecting, your overall yield will be 0.001%. So you will even be challenged to find the final product in this nasty mixture. If you go to 90%, 95%, at 95 you're at about 8% total yield. At 99% you're at 60% yield. Now if you go up to 99.5%, it would be 77%. You see, 0.5% yield increase in a single step gives you 17% increase in the total process. This improvement is what's really driving prices, because your yield is improved. And this is also why I mentioned the supply of starting materials before. That's so important. We save a lot of money by buying the most expensive starting materials we can get. Offering the right capacity is an absolute key element. And the flexibility to choose between different capacities is a main differentiator between doing it yourself or working with a third party. The third party will often have more than one production unit, while if you're doing it yourself, you have to choose one size, because very often it's dedicated material. And in this booming market of peptides and oligonucleotides, capacity is an issue, especially if it comes to large volume capacity. I therefore want to show you in the last part of the presentation what Bachem is doing, how we're investing into capacity for the future. But I also want to comment on why Bachem, the leading manufacturer of peptides is now also aspiring to have a top position in oligonucleotides. There is a large unmet need for the production capacity, especially for large quantities, also in the oligonucleotides. A new product branch that offers new therapies to a lot of patients. But the main reason for us to step into this is that the manufacturing of those two types of products is very similar. So the upstream chemistry is different. However, it's the same principle. You couple one of the elements to each other, very often on solid support. Downstream, when you cleave it from the solid support, you're deprotecting it, same thing. Cleaning it at large scale, that usually means HPLC cleaning. And very often lyophilization is method of choice for isolation. So you see a lot of similarities in those two processes. And talking of investment, Bachem is investing strongly in capacity. What you see on this picture is our site in Bubendorf. The red building on top is the ongoing expansion we call Building K. This will be our largest tied manufacturing facility, adding more than 15,000 square meters of space and will include the latest technology. Groundbreaking was in 2021 and we're expecting it to be operational in 2024. While Building K is not even finished, we're already planning the next step. We're planning for an additional manufacturing site in Switzerland, and we want to have this operational by the end of this decade. We make this investment so you can focus your attention on the clinical development of your product. To summarize it, the hurdles for manufacturing a product yourself are often high and need large upfront investment, especially for complex products like peptides and oligonucleotides. Working with a CDMO gives you much more flexibility. And even the cost is usually better if you work with a specialist, despite the fact that they also want to earn something in the process. The key elements for choosing a CDMO, in my opinion, are innovation and cutting-edge technology for optimal processes. You want operational excellence and good planning for flexibility and speed. You want top quality for reliability. You want them to focus on the customer for understanding their needs. And you want the right people to offer all this. A good CDMO doesn't just sell you the product. They support you with your product and make your product a success story. I hope at the end of this talk, I was able to convince you that working with a CDMO instead of doing it yourself is the right choice. I thank you a lot for your attention. And before answering your questions, I hand over to Daniel because we have one poll question to ask you as well. Thank you.
Hey, thank you so much, Tujo, for your for your presentation, which we recorded, as was mentioned earlier, a little bit ahead. And before we go into the Q&A, here's myself, here's Tujo, live with you. And we would love to ask you all a question. We've got about 80 participants in this call, by the way, who've held on throughout pretty much all the presentations. So that's very, very nice. Seems this is an interesting subject for everyone. So our question to you to kick this off, what is your key selection criteria for a CDMO? You can make multiple choices here. Please tick one or multiple boxes as to what you find most important for a CDMO. We'll collect some of the data and play that back to you in just a moment. While we wait for this, I'll give you a quick note on how to ask questions already. So we have a Q&A function in Zoom. If you scroll down to the bottom of your screen, there should be a button with Q&A, and then you can type a question. We already have some coming in, which is great. And here are our results. We have a winner here. 74% say key selection criteria for a CDMO is quality. Price is a runner-up, strong runner-up with 64%, and then 48%, about half people also say large-scale capabilities, almost as much then as analytical capabilities. Cooperative mindset is also one, the culture piece, very nice to see that. And then some notes on supply chain and sustainability. So we're coming to the live Q&A, and please keep your questions coming. Write them into the Q&A. I will pick the first one here that we have from James Hay for Tushio. And Tushio, I will read this question so everyone knows what it's about, and then you can get to answering it. This one is about technology. James Hay writes us, what key technology advances are being made in peptide manufacturing? It used to be liquid phase synthesis, then the market moved to solid phase synthesis. Now seeing other innovative solution phase approaches, what does Bachem offer that is differentiated in this area? Tushio, please share your thoughts.
So I guess it really depends on your project. If your project is very large scale and a very short peptide, the answer might still be liquid phase chemistry. And the negative point is if the product is too long, this will take very long time. We still got old products, 50 mirrors that have a lead time of a year or even more to be prepared. So there we're switching actively to SPPS. Now, what we're doing technology-wise for the LPPS, we're switching to production facilities that are completely closed. So that's good for health, safety, environment. So we need to have the environment protected, but also the people protected. So that's things we're still investing even in this older technology. And as mentioned in the talk before, it's really a matter of choosing the right one for the product. And if the quantities are large, and if the product permits, new technologies like this in-between, this go-between technology from Chitsubo, where you have still kind of a support where you couple your molecule to, but a molecule that is then kept in solid phase, in liquid phase, that's kind of a go-between those two things. Of course, we're also investigating other things like enzymatic processes, but so far the advantages there were not really big enough to really promote the switch from the SPPS, because there the processes were really improved to a way that it's very hard to get something that still is better.
Okay, thank you very much, Tushio, for that answer. And we have the next question. This one comes from Yogesh Sanghvi. I hope I pronounced your name right. And the question is about continuous chromatography. So Yogesh writes us asking, what type of cost and time saving do you see when continuous chromatography is implemented?
I'm not the absolute specialist on this, so really go watch that webinar. I'll try to answer as good as I can. We have the possibility, of course, to get to a higher quality of the product. So if this then means in the end, and that's really project dependent, that you can go from a 3D to a 2D process, this might save you something. Your solvent utilization is reduced sometimes by 50%. So my guess would be for a large enough project, you might end up with cost saving on that step of around 30%.
Okay, thank you, Tushio, for that. And just picking up again, we do have a webinar on the topic, so check that out on our website. You'll be able to find it, or on our YouTube channel recording. Next question. This one comes from Nishat Soni. This one is about, this is an interesting one. This one is about regulatory challenges depending on the method of manufacturing. So question from Nishat is, are there greater regulatory challenges for peptides produced by synthetic methods as opposed to biological methods? Is biologic peptide production considered at Bachem?
In the beginning, I would have said, yes, there is different challenges because this draft FDA guideline that was put into place like half a year ago was especially targeted at synthetic peptides that were replacing recombinant ones. However, what we see right now is that this is basically being applied to everything. So even if you're substituting a synthetic peptide with another synthetic peptide, it's basically the same thing. The challenge of this draft FDA or this FDA guideline is that it requires you to have all the impurities below a level of 0.10%, so very low impurity levels. And if you want that high purity, very often that means you're cutting your HPLC fractions very tightly. I could imagine that some recombinant products are easier to filter out because your impurities might be less similar to your final product compared to synthetic ones. However, you still have degradation products, etc., that are also then very, very close. So you probably end up with quite similar methods that you need to purify those products, and the same regulatory requirements apply. When we look at the first FDA guidelines regarding the first oligonucleotides, so what do you need to do there? We see that this one really mentioned finished dose producers will get away with a bio waiver, and the challenges for the API manufacturers, those hurdles are really, really tough.
Okay. I see. It's nice to see we really have an active set of people asking questions here. Yogesh, by the way, says thank you for your response to the question earlier on continuous chromatography. Here's the next question. This one comes from Norman Miller, and he is asking us this one is about cost in relation to size and technology. So Norman Miller asks, for a small peptide, for instance, with 4 amino acids, how much more expensive is molecular hiving as a green technology compared with standard manufacturing?
I guess in the development of the method, this will be more expensive. I could imagine that actually then manufacturing it won't really play that big role because you're talking about really usually then larger volumes. And there you will have the same prices for the starting materials. You will have the same prices for the cleanup, etc. If you're able with the molecular hiving to do this in a one-step process, this will differ somewhat. Former is either you do it consecutive or then it's a synthesis that you pair 2 and 2 and put them together. So at least you have 3 steps there where you need to do a workup in between. So you might end up with quite similar prices in the end. It might even be that molecular hiving offers you a competitive advantage on the manufacturing side. Of course, you need to pay for the support as well. So yeah, really will depend on the process. My guess is that in many cases, you might even end up cheaper with molecular hiving, but the upfront investment is higher.
Thanks for that response to Norman's question. And so, yeah, we still have the vast majority of people who joined us at the start on this call. So it seems you're at least interested in responses. Do let us know if you have any additional questions. I would put one now to you, Tycho, as we're seeing whether we'll get initial questions through the Q&A. But one thing that struck me when you spoke about oligonucleotides was the fact that these are going increasingly into head-to-head trials with other medicines. And I was just wondering whether you could say a little bit more on that point, what that means for companies to consider, what they need to consider if, what is the change sort of from not going to head-to-head to going to head-to-head settings for companies when they're looking for CDMOs?
I mean, basically, if you're treating a patient who was not treatable before, very often that's the case in orphan disease, where the oligonucleotides really opened the door to completely new therapeutic options where no other existing therapy was possible. So in the end, you end up with a discussion with the payers on, can you treat this patient, yes or no? What's the value of this life or this quality of life that I'm adding? This discussion will be a completely different one if you enter, like it is done at the moment, a field like hypercholesterolemia, where you have patients that have a broad range of possible treatments already, like statins that are there for 20 years now, very well established, very effective, and very genericized. So those prices are really low. And when you come now with a new therapeutic option, well, you only need to inject it once every half year. Compliance is a huge issue with those patients, but nevertheless, you need to compare your treatment also from the cost perspective to the existing one. And you need to show that, it's worthwhile to allow a certain price for this product. This, of course, then also pushes the price pressure on the whole supply chain. And you also need to show in clinical trials, not just that it works, but you also have to show it works better than what's existing and it's better tolerated, etc.
Okay, thank you. We have another question here, again from Yogesh. This relates to green chemistry and oligonucleotides. Rather technical piece. Let's see what you think of it. What have you done at Bachem related to the use of green chemistry for oligonucleotide synthesis? Something you can respond to.
With oligonucleotides, we just started. We're playing in this field for a longer time. With the present technology, we just about have the first GMP batch that was done some months ago. So there, we're still in the beginning. However, basically the same principles apply. So continuous chromatography, for example, that's something that you can use for peptides, but you can use it as is one-to-one also for oligonucleotides. So those kind of things that you're investing in, that's transferable one-to-one also on oligonucleotides. And we're heavily investing there also in innovation. So we're trying to bring our expertise from the peptide manufacturing also to the oligonucleotide side and try to improve this there as well. So again, utilization of solvents is a thing. So there we see some potential, but that's not yet in a finished process.
Thanks to all participants. So if you still have questions, please do use the Q&A function. We still have some time and scope to respond to them. I've got another one right here coming in from Nishant. And this one is about sustainability, maybe more in a broader sense. So what are the sustainability challenges for Bachem considering your large manufacturing capacities with current and future requirements of therapeutic peptides. And then there's, I guess, a second part of the question, which I'm not sure it's fully related, but that's what happens if other treatment options like mAbs and gene therapies become a bit more preferred. So first part here, what are the sustainability challenges for Bachem considering large manufacturing capacities with current and future requirements in peptides? And then second part of the question, what happens if other treatment options like mAbs and gene therapies become a bit more preferred?
One thing, for example, if you do HPLC or UPLC in your lab or in QC, it's easy. You've got a 5-liter bottle of your solvent on top of your machine. If you're doing this with a 1-meter column, at the moment, that's the latest pictures on our site, because the big tanks are being installed in the cellar of this new building. So we're talking about huge quantities of solvents that are being used. So whatever you can reduce there will be a big thing. So with continuous chromatography, that has a huge impact, because if we're talking about large scale production for those products, cleaning them up to a product that is afterwards acceptable, that requires a lot of organic solvents that then end up in the waste. So again, the second thing is, how do you treat your waste? And their regulations in Switzerland and California are among the most stringent you find on this planet. So we already comply with the regulations that are the most severe, and we try even to surpass them. And we have very ambitious goals for green technology, and we want to be carbon neutral also. And with this solvent utilization, that's something that is quite challenging, but that's something we're actively working at. And then the second part was regarding the therapeutic modalities. So that's absolutely clear. If a company sees the option to treat a certain disease, it doesn't really matter what kind of technology is behind it. You just want to treat the patient as good as possible. However, I'm not that convinced that actually monoclonal antibodies are really the answer, because with those biologics, very often you have immune response that is not really captured in the clinical trials. So in longer term treatments, very often you see that they're less well tolerated, and that an immune answer is being built up. So we also see, like in the GLP-1 products, their dulaglutide was a monoclonal antibody, but the products that now enter the market, they're not additional monoclonal antibodies. So the first one was exenatide and liraglutide, and then came the monoclonal antibody, but now it's again peptides that enter this market. So the new technologies there are the peptides and not the monoclonal antibodies. And with gene therapy, I think this is a very expensive therapy, and it really depends whether or not in a certain disease the outcome really justifies this high expense. So I think it's a case-by-case thing, and in the end, what's best for the patient is probably that mean that should be pursued.
Sergio, thank you very much. That's almost a great closing statement, actually. What's best for the patient is the path that should be pursued. I don't see any more questions here in the Q&A function. So seems to me, I'm also seeing that I think people are gradually dropping off after sticking with us for pretty much all the call and all the Q&A. So for those of you who are still here, we're just a pretty good big bunch. If you want to learn anything more about Bachem, please do check out our new company brochure. We just updated a completely new revamped version. And you'll be getting the link to that and the slide, a summary of these slides in an email Thank you very much for joining us here today in this, I would say, very insightful presentation and Q&A session with Tuchel. If you have any further questions, our teams are always there to respond to them and get in touch. Obviously, if you want to do business with us, we would love that as well. So as we close this call, I bid you all a good night and or a good day, depending on where you are in the world. And the last note I have in our Q&A chat is from an anonymous attendee saying, thank you for a very informative session. I will look forward into your other webinars. So that's also a lovely quote here. With that, see you in our next webinar, or maybe if we're lucky in person, and have a great day or night. Goodbye.